We all have noticed the cottony growth on a stale piece of leftover bread. It happens with many food items when they're left open, especially in moisture. But have you ever wondered why? What exactly gets this cotton-like substance here? What is it made up of? Well, the cottony growth is nothing but a set of live cells. Yes, these are living cells, and this fact is more than enough for us to study them. By now you must have guessed the name of these organisms. Yes, I'm referring to the fungi. Fungus is usually a broad term used to cover many organisms together. The group consists of microscopic as well as macroscopic organisms. What do we mean by this? Microscopic means those which can only be observed under the microscope. So the cottony growth that you find on stale bread can be included in this category. On the other hand, macro means huge. So those which are big enough to be observed with naked eyes are macroscopic organisms. Which organisms can we consider as an example of this type? Well,
Sir,
mushrooms and puffballs can be included in this category.
Sir, can you pause once?
Yes.
Yes,
Just take a
sir.
screenshot.
I'll copy it, sir.
I will give you idea about that. Don't worry, beta.
Let's talk about the microscopic types first. These being too small, what do you think will be their structure? Do you think they will always be unicellular? Well, not all. Only a few types like yeast are unicellular, whereas others, in spite of being microscopic, are multicellular in nature. The unicellular forms exist as one single cell which performs all the activities efficiently. On the other hand, the multicellular ones have bodies comprising of many cells that coordinate to bring about smooth processes. The multicellular body usually consists of the stalk-like structure
beta?
and a cap on top of it.
What did you say?
Nothing, sir. Nothing, sir. My sister
okay.
is disturbing, so.
This cap comes in various forms. Some of them may have a bag-like structure. Others may have umbrella-shaped structure. Both these are dedicated to a special role. They contain spores of the fungi, which can be dispersed to give rise to a new fungal bodies on germination. Now moving to the stalk. This usually consists of long, slender filaments. These filaments are the cells that make up the entire fungal body. They form a dense network together. So, are these filaments given any specific name? Scientifically, they are addressed as the hyphae or singular hypha. These threads get interwoven to form a network. This structure is what we call as the mycelium. So can we say that the mycelium is a collection of hyphae? Yes. And can we also say that the fungal body is made up of mycelium? Absolutely. Thus, this stalk that we can see here and also these root-like structures are all mycelium made up of hyphae. Now one more important question that pops up here. Are these hyphae made up of one continuous tube? Or they are made up of smaller sections? Let's zoom in further to know this. Some fungi have hyphae that have partitions. These partitions, as we can see here, are called as the septa or singular septum. So such types of hyphae are called as septate hyphae. Now can you guess the next type? They will be without the septum. In many fungi, you find no partitions in the hyphae. These are called as aseptate hyphae. Another name for such type is cenocytic hyphae. Now you may wonder as to why the structure of these hyphae is so important to us. What could be their possible function? Well, apart from forming the entire body of the fungi, these help in many ways. Hyphae are ideal for absorbing nutrients from the surrounding food source. Precisely, the tip of these hyphae release enzymes that bring about digestion of food outside the body. After breakdown, these nutrients are absorbed directly by the hyphae and are passed to various parts of the fungal body. If the hyphae are aseptate, then the nutrients can be directly passed. However, in septate hyphae, the case is different. The septa have small pores which allow passage or even exchange of substances. The hyphae are also known to contain cytoplasm and a few important organelles. And most important is the presence of nuclei inside them. Thus, the genetic material of the fungus is locked in these hyphae. For all these reasons, the hyphae are important to us while studying the fungi. Well, this was the multicellular structure of fungi. But this is what we observe under the microscope, right? So will the huge macroscopic structures that we see in case of mushrooms have a similar internal structure or something different? Think for a moment. Well, astonishingly, these also have a similar internal structure. So the body of the mushroom will also be made up of mycelium. Similar to the microscopic ones, even this mycelium will be made up of interwoven hyphae. So we may find slight modifications in these different types of fungi, but the basic structure remains the same. Well, you may wonder as to why we need to study about these fungi. What is their importance? Let me explain. There are several important roles that fungi play in ours as well as other organisms' lives. Some of them include forming a symbiotic relation with plant roots. Symbiotic means a mutual give and take relation. So both organisms, the plant as well as the fungi, are benefited in some or the other way. Secondly, helping us with many medicinal important products. Do you know any such product obtained from fungi?
An antibiotic
Of
drug.
course you do. You are familiar with the antibiotic penicillin?
Yes.
This is obtained from the fungi of genus Penicillium.
Discovered by Alexander Fleming.
Similarly, many of the foods that we relish, including bakery items and cheese, are all a gift of various fungi to us. So don't you think studying them is important for us? That is the reason why we have a complete branch of microbiological studies dedicated for studying fungi exclusively. This is called mycology, where mycos is fungi and logos is study.
Will there be questions about the structure of fungi?
Structure of fungi?
Fungi.
Okay. We will discuss from the notes now, beta. Whatever the structural part we have, whatever I have like shown you in a video, it's like quite you can say detailed, beta. We do not have that much of detail in our
Sir,
syllabus.
for exam only till here, sir.
Yes, yes, beta. Okay, yeah. So let's discuss about the fungi from nodes. So they are a group of single cell. So single cell we discuss which one? Yeast, right? And they will be multi-cell.
Yes,
So
sir.
you can say mushroom. And we eat mushroom. My favorite, bro. You know, mushroom is my favorite. Yes. Okay. And it is very full with protein, beta. Okay. So fungi may be neither microscopic. Okay. Either they may be either microscopic and unicellular, like yeast we have, and molds, or macroscopic means they have very large structure and multicellular. Like example, mushroom and toadstools. Okay, you will find those on the rock or maybe on other plant.
So the saprophytes.
Saprophytes. Yeah, they are called as saprophytes. Yes, because they do depend on the dead organic matter of plant and animal. Okay.
Okay, sir.
All fungi lack chlorophyll, so they do not perform photosynthesis. So they do depend on other organisms for their food. So how they do depend on other organisms? They do
For
depend
example,
on
the
the
bread.
dead organic matter of plant and animal. Okay.
Yes, sir.
Okay. And derive their nutrition from the decaying matter. So they used to decay the matter and they absorb their nutrition from there.
Okay, sir.
Okay. Okay, let's discuss, get it right. Now, all fungi and algae are microscopic. Some of them are quite large, like mushroom and seaweed. So, seaweed is the algae. Okay, and mushroom is the fungi.
Okay, sir.
Okay. Now, let's discuss about the algae. So, they are the group of simple plant like organism that contain chlorophyll. If they contain chlorophyll, so they do perform photosynthesis. Beta. Photosynthesis, right?
Yes, sir.
If they do
Yes, sir.
like perform photosynthesis, so they do prepare their own food. Okay, and they do not depend on other organism. Okay, they may be unicellular. So in unicellular we have chlamydomonas or multicellular. So multicellular like seaweed we just discussed. Okay, some unicellular algae also exist in colonies like caldera and diatoms. They are the single cellular, but they generally present in colonies, beta. Colonies like many unicellular present together like this. Okay. Okay, so this is about algae.
Sir, can you pronounce this unicellular one, sir?
Okay, that one,
Yes, sir. CH.
caldera. Caldera.
No, sir, not that one, sir. The unicellular.
ALGE, algae.
No, sir. Sir, they may be unicellular. Example,
Chlamydomonas.
chlamydomonas.
Yes.
Sir, 1
Chlamydomonas.
minute. Sir, I'll copy the spelling, sir.
Okay. Chlamydomonas or multicellular seaweeds.
Okay, sir.
Okay. Now let's discuss about virus. Viruses are like interesting study of virus. Why, beta? Because they are kept between living and non-living things. Okay. So first on Earth we got non-living things, right?
Yes, sir.
Yes, sir. Yes,
After
sir.
non-living, viruses are formed virus. And after virus, bacteria are forming living things.
Sir.
Huh, beta? Okay.
So yesterday we discussed that bacteria are the first living.
Yes. So yeah, it was a living thing, no. But virus is between living and non-living things, beta.
Okay, sir.
So virus are, that's why I told you virus studies become interesting. Why? Because if virus are present outside the body of the organism, so they are dead. But if they present inside the body of living organism, so they are alive. Okay, that's why viruses are kept between living and non-living things. Okay, so they are so
Okay, sir.
small, they that they cannot be using, we cannot see with the help of compound microscope also. So we do require electron
Electron
microscope in order to see them. Yes, viruses are hard to classify as living or non-living. Okay, it is very difficult to classify them whether they are living or non-living. Okay, why? They shows no sign of life when they are present outside the body alone. Okay. However, they reproduce inside the cell of organism such as plant, animal, or bacteria. Okay. If suppose they are present inside the cell of plant or animal or bacteria, they start reproducing. Okay, means they are living things. So they are present in a body, cell of the body of any organism. So they are living. If they are present in outside the body of organism, then they are non-living.
Okay, sir.
TMV, tobacco mosaic virus. Okay, it is present in tobacco. Beta, you know tobacco. It is present in cigarette, gutkhas, right?
Yes, sir.
So this tobacco is nothing but derived from the plant only, right? And that plant is getting infected by tobacco mosaic virus. That tobacco
Sir,
mosaic
is it
virus.
harmful? Is it harmful?
Yes.
Okay, sir.
Yeah. So that tobacco mosaic virus secretes some enzyme which present in a tobacco leaves, and that tobacco leaves used as a drug. Drug means it creates addiction better. Okay. Tobacco mosaic virus and human immunodeficiency virus (HIV) are the example of viruses. Even we have one more, COVID-19, worldwide famous virus you can say, are widely feared when flu and swine flu are also caused by the virus. So flu and swine flu and upper and lower respiratory tract infection is caused by virus only. Okay.
Yes, sir.
Sir, 1 minute,
Sir?
sir.
Okay.
I'm copying.
Sir, is yeast a fungi or bacteria?
What beta? That one?
Is
Fungi.
yeast a fungi or
Yeast is
fungi?
fungi, unicellular fungi.
No, it is a fungi, right?
Yes.
Okay, sir.
Unicellular, not multicellular, it is unicellular. Huh? Uni means one. It is made up of single cell.
Okay, sir.
Any doubt till here?
Sir, what is sir, how many variants of flu are
How many variants of
flu?
flu? Okay.
Like
See,
do all the
all the flu
variants
they
of
are caused by viruses only, but virus may be come from the different source, like from monkey, from bird, from other animal like pig, horses, cat, etc. Okay. So generally
Okay,
in
sir.
flu, we are getting cough, cold, and fever. Okay, so till here, any doubt, beta? In virus, algae, and fungi of protozoa, any doubt here?
sir? So there is no useful virus.
No doubt,
Useful
sir.
virus.
Sir, I
Beta?
think there is more 10 minutes.
10 minutes, 25. Oh no, we have only more 5 minutes. Any other lecture?
Sir, we can
will
have quiz.
Yeah, we can have quiz on that.
Okay, sir.
I finished. Sir, I finished.
You finished? You can leave if you finished.
Okay, thank you.
Everyone test is finished.
Yeah, done. I'm
Okay,
leaving.
then you can leave. Bye, bro.
Bye, good night.
Pranav, is your test completed?
Just one more question.
Yeah, okay.
Okay,
Harshit, yours?
yeah, done.
You can leave, bro, actually. You can leave. You can leave and write, okay? Bye.
It's over. I finished it.
Yeah, okay, bye.